Hello and welcome to today's special GeneWest Week webinar, Rewriting Natural Decoding Rules by De Novo Genome Synthesis. I am Dr. Sumit Kumar, Senior Product Manager at GeneWest, and I will be moderating today's webinar. I am very pleased to introduce today's presenters, Dr. Kaihang Wang and Dr. Julius Fredens. Dr. Wang is currently working as Senior Investigator Scientist at Dr. Jason Chen's lab at MRC Laboratory of Molecular Biology, also known as LMB. He spent a year at Peking University in China before moving to UK to finish his undergraduate studies at University College of London. He then pursued his doctoral studies at MRC LMB in Dr. Jason Chen's lab, expanding decoding capacity of orthogonal translation machineries. He became a junior research fellow at Trinity College, Cambridge University after his PhD graduation, then worked as a career development fellow at LMB. He continued to work at LMB in various different roles, including investigator scientist and then senior investigator scientist, while working on de novo genome synthesis and engineering in E. coli and beyond. Dr. Fredens is a postdoctoral fellow at Dr. Jason Chen's lab at the MRC Laboratory of Molecular Biology. He did his undergrad and PhD at University of Southern Denmark, where he developed a method for quantitative proteomics in C. elegans and used it to study regulation of metabolic pathways. After graduating, he joined Dr. Jason Chen's lab at MRC LMB, first as a postdoctoral fellow of the Danish Council for Independent Research, and later as Lundbeck Foundation's international postdoc. He is currently working on genome synthesis and engineering in E. coli and beyond. If you have any questions for Dr. Wang or Dr. Fredens during the webinar, please enter them into the Q&A section and we will try to address as many as possible at the end of the session. With that, please welcome Dr. Kaihang Wang and Dr. Julius Fredens.
It's our great pleasure to be part of the GeneWest webinar. We would like to share our past passion and also future vision with the global audience. The question we are trying to pursue actually has a really ancient root. Since the beginning of life, one thing has been said, which is a central dogma, in which DNA makes RNA makes protein. The genome not only contains all the DNA that contains all the information of life, the genome also contains an embedded set of decoding rules, which dictate which one of the 64 triplet codons encode which one of the 20 natural amino acids. And here are the 20 natural amino acids. Altogether, they do offer rather diverse chemical possibilities on their side chains. However, if we compare the limited 20 possibilities with the unlimited chemical space, unfortunately, the 20 natural amino acids is rather conserved in its chemical and physical properties. The idea we would like to pursue is potentially, if we could de novo synthesize a new genome, then we can redefine a new set of synthetic decoding rules embedded in that new synthetic genome, such that we can fundamentally go beyond the limitation of the 20 natural amino acids and expand the translational decoding capacity way beyond and into unnatural amino acids, which potentially will give us ability to venture into lots of exciting applications, such as antibody-drug conjugations and incorporating post-translational modification directly onto proteins. The exciting question we want to really ask is thinking about nature. Evolution made us from nothing based on just 20 natural amino acids. Potentially, if we could really free evolution from this limitation of the 20 natural side chains into the unlimited possibility of unnatural amino acids, then where evolution can lead us to, and also more importantly, where bioengineering driven by human determination can actually take us to. The fundamental question is, how do we actually make and design a new genome that allows us to pursue this dream? The idea is by looking into the natural decoding rules, right? This is embedded natural decoding rules using serine as example. And there is quite a degree of redundancies. For example, there are 6 serine codons encoding the same serine amino acid. What we want to do is we want to first reduce the natural decoding redundancy to some extent and free up certain codons from the natural decoding triplet decoding boxes, and thus use the freed up decoding capacity to actually reassign to encode unnatural amino acids with 100% efficiency. This requires us to remove the target TCG and TCA codon in this example through the whole genome and also their cognate RNAs. Because of the density of the TCG and TCA across the whole genome, the only realistic way to actually achieve this goal is through the novel genome synthesis. Then the question becomes, how do we actually devise a way to actually truly make a synthetic genome purely based on synthetic design from synthetic materials?
The most impressive experiment like this in the past has been an experiment where the whole genome in the microorganism mycoplasma has been replaced with a synthetically chemically synthesized genome in one go. This is very impressive work, but it has some limitations. First of all, we have to have a genome that is so small that we can actually handle it, and that is not the case for very many organisms. Second, the organism in question here has to be able to take up such a big piece of DNA in one go. And that is the case for mycoplasma, but not the case for very many other organisms. And thirdly, if you synthesize and massively redesign a big genome like this, then you have to expect some of the design features to be disallowed by the organism, and if you replace the whole genome in one CGR at one time, you have a difficulty figuring out exactly which part of the genome that fails to make a viable organism afterwards. What we are suggesting to do instead is to take E. coli and replace the genome step by step in small bits, partly because we can't get a whole genome in at once, and also because we cannot, the genome in E. coli is much, much bigger than in mycoplasma. So by taking small pieces at a time, we hope that we can synthesize bits of the genome one by one, and every time we make a design that is not allowed by the organism, we can tell exactly where it is, we can fix it, and then we can move on. It looks a bit more tedious this way, but we actually believe that this is a more sustainable method.
Now the next question becomes essentially how can we introduce synthetic piece of DNA reliably into the genome to replace the genomic fragments. The classical way of introducing synthetic DNA into the genome is through lambda-right recombination in E. coli. Right, in the lambda-right classical recombination, you first have a piece of linear double-strand DNA, typically generated through PCR. You transform the synthetic DNA into the cell, and at the same time, undergoes recombination. You select for the correct clones, the correct recombinant by select for the gain of the coupled positive selection marker, in this case the plus here. And the integration of the synthetic DNA is located into the right locus, supposedly driven by the homology based on the homology regions. This classical recombination has been the tool to integrate synthetic DNA into E. coli genome and many other systems as a textbook method for decades. However, it does come with several intrinsic limitations. And the first limitation is the efficiency is actually really, really low. This is because for the classical recombination, in order for this to be successful, the transformation and recombination has to happen at the same time. However, none of the two is very efficient. So the possibility of two low efficiency events happening at the same time is extra low, making the efficiency is actually limited to typically a few hundred clones per experiment, and with actually short synthetic DNA. And making things worse, as the size of synthetic DNA gets longer, the efficiency of the transformation drops down really, really quickly, such that for any synthetic piece longer than 10 or 20 kbs, you don't really have any successful integration anymore. And also, as the set of integration is purely a function of the homology regions in the classical combination, the local specificity is rather limited with the classical approach. And in order to actually have a method that can truly allow us to replace long piece of genomic fragment with synthetic DNA, we really need to overhaul this classical method and address all the limitations before we can move forward. And in order to address basically all the limitations of the classical recombination, we actually invented and developed a new method which we call replicon excision enhanced recombination. In this, or REXA for short, in the REXA strategy, basically we fundamentally solve the problem of the limited efficiency and the length dependency by decoupling transformation from recombination. The way we do this is by putting the synthetic DNA on a stable episomal replicon, in this case a bacteria artificial chromosome, or BAC. And when we transform the BAC into the bacteria, we pick one individual colony and grow it up. Then basically in that population, every single cell will contain the BAC, which contains the synthetic DNA. Then, from that stable population, we excise the synthetic cassette from the carrier BAC by using CRISPR-Cas9 cuts. In the REXA2 setups, there are two CRISPR-Cas9 cuts, flanking the synthetic cassettes there and there. In the REXA4 cuts, besides the two cuts flanking the synthetic cassettes, there are two additional cuts on the E. coli genome, flanking a first double selection cassette -1+1, which we previously integrated to mark the locus of integration. And after the CRISPR-Cas9 cuts, essentially we do recombination. Through the REXA2 strategies, the recombination can happen through strand invasion, while through the REXA4 strategy, the recombination can happen through strand annealing. In order to overcome the limitations of locus specificity, we actually, based on our design, devised this double selection strategy, in which we select for the gain of the synthetic piece of DNA by the gain of the coupled second double selection cassette by the selection of the gain of the second positive selection marker +2, and also the loss of the original genomic fragment by the selection of the coupled first double selection cassette -1+1 by the selection of the loss of the first negative selection cassette -1. By doing the double selection of the gain of +2 and loss of -1, we ensure every single cell that survived the REXA strategy contains the synthetic piece in the defined locus with 100% specificity. And because fundamentally based on REXA design, the transformation and recombination are decoupled, the efficiency is really, really high. With REXA2 strategies, you have tens of thousands clones per experiment. With REX4 strategies, you have tens of millions clones per experiment. And also more importantly, because the transformation is decoupled from recombination, and actually in the REXs, both ends of the synthetic cassettes function independently. The integration efficiency is actually designed to be independent of the size of synthetic DNA, and experimentally, it truly is completely independent of the size of synthetic DNA. You have that tens of thousands clones efficiency with REXA2, regardless whether you are integrating a 2 kbs or 100 kb synthetic DNA, and the same goes for the REXA4. You always have the same tens of millions clones per experiment efficiency, whether it is a 2 kb or 100 kb integration. We first implemented the REXA strategy to insert long piece of synthetic DNA into defined locus of the genome. We first mark the locus of integration with the insertion of a first double selection cassette -1+1. Then we synthesize the 90 kb of synthetic DNA and couple that with the second double selection cassette -2 and +2. Then basically we do the REXA strategies, either a REXA2 cut just flanking the synthetic DNA, or REXA4, in addition to cutting flanking the synthetic DNA, we also cut on the genome flanking the first double selection cassette. Then through the REXA2, we generate a linear piece of synthetic DNA 90 kb long, with double strand breaks there and there. In the REXA4 strategy, we generate essentially 4 double strand breaks there on the synthetic cassettes, and also 2 double strand breaks on the genome, which allows the recombination to happen through strand annealing pathways. And then we select for the gain of +2 and loss of -1, which in either REXA2 or REXA4 setups lead us to the same final product, which is integration of the 90 kb synthetic DNA into the right genomic locus. And over here, in order to mark the whereabouts of this 90 kb synthetic DNA, we actually spread 5 LUX genes across the entire length. The 5 LUX genes together, they make up the bioluminescence operon and basically make the cell bioluminescent. You need all the 5 genes there to be actually to light up the E. coli cell. And here is our experiment result for REXT4 strategy. For REXT4 setup, integrating 90 kb of insertions. This is a 10,000-fold dilution plate. As you can see, even after 10,000-fold dilution, you still have thousands of clones on the plates. And when you turn off the light, actually every single cell lights up as confirmed by this superimposing as confirmed by this particular image superimposing the dark image and the white light image. And this really confirms essentially with 90 kb insertions following the REXT4 strategies. You have tens of millions clones per experiment, or 100% successfully integrate the 90 kb synthetic piece. And I mean, the insertion with Vexer is really reliable and straightforward. However, in order to actually convert a wild-type genome into a synthetic genome, we actually also need to do replacement. And we can also use Vexer to successfully do long piece of replacement, following the same strategy. And we first mark out essentially the starting as a starting site of the replacement with the integration again with the first double selection cassette -1+1. Then we synthesize a 100 kb synthetic DNA designed to replace the first 100 kb of the genomic fragment and couple the second double selection cassette with that and put that on the back. Then basically we do the Vexer strategy and select the gain of +2 and the loss of -1, which lead us to the successful replacement of the first 100 kb genomic sequence with 100 kb synthetic DNA. One important design feature of the Vexer systems is the product of the first step of Vexer will serve as a template for the next step of Vexer right away. And basically, based on the product of the first step Vexer, we can provide a second 100 kb synthetic DNA designed to actually replace a second 100 kb genomic fragment. Now, actually, the second 100 kb synthetic DNA are coupled with the first double selection cassette. And then we do Vexer again. This time, select for the gain of +1 and the loss of -2. This way, we actually, this is basically lead us to the successful replacement of the consecutive second 100 kb genomic fragment with synthetic fragment. We can basically expand this principle into the entire genome. And basically with the fact that the E. coli genome is around the size of 4.5 megabase, and with the fact that the back can carry up to 300 kb of synthetic DNA, we envision to be able to replace the entire E. coli genome into the Novo synthesized one using synthetic DNA in 13 to 15 steps. We call this strategy genome-wide stepwise interchange synthesis. So, based on Genesis, we truly now have a viable way to actually go from a wild-type E. coli genome and stepwise convert that into eventually a de novo synthesized genome. Then the question becomes, how do we actually want to design this new synthetic genome to actually achieve our final goal, the goal of expanded decoding capacity into incorporation of unnatural amino acid?
So as Kai said in the introduction, the genetic code is degenerate. And for example, for the serine codons, we have 6 different codons encoding the same amino acid. The strategy for serine would be to choose 2 codons, for example, TCG and TCA, that are highlighted in gray here, and delete them from the whole genome and replace them with one of the other synonymous codons. After removing all the TCG and TCA codons, we can remove the 2 tRNAs that are decoding these particular codons. They're here are highlighted in gray as well. And after removing the tRNA, we have an organism that doesn't contain the codon and doesn't contain the tRNAs for these particular 2 codons. We actually have a blank spot in the genome here. And now we can subsequently introduce we can introduce new proteins that contains these 2 codons and the new tRNAs, so we can introduce unnatural amino acids in a clean and undisrupted way. But the question is still, how do we choose which codons to replace with each other? Because we know that synonymous codon choices is very important for protein expression and other functions in the genome. And we could, of course, just try and see with all the different possibilities what works and what doesn't work. But the problem here is that there are way too many combinatorial options. So the solution to this problem, as we see it, is to try to make a model that predicts which codons are likely to be replaced with each other. And then when we have narrowed it down to a couple of different models, we can simply just test them in the setup we just have described. Now, when we look at the serine codons, we want to replace the TCG and the TCA codons. And we came up with 3 different models that predicts different ways of replacing. One model suggests that both TCG and TCA should be replaced with AGT. A second model suggests that they are both replaced by AGC. And a third model suggests a combination of the 2 previous ones. We want to do a similar trick to the leucine codons. Here we also have 6 codons encoding the same amino acid. And if we focus on the 2 codons TTG and TTA, we have 3 different models that predict different recoding patterns. Finally, we looked into alanine, where we have 4 codons encoding the amino acid. And if we replace 2 of them, GCG and GCA, we end up with 2 models that are suggesting 2 different replacing strategies. Now we have an idea of how we want to replace the genome, the codons. Now we have to figure out how to test it. So we looked through the genome and found a specific region where there are a lot of these particular target codons in essential genes. This region is very dense in essential genes, and it has a number of other features that makes us believe that this is the most difficult region of the genome to replace or to recode. So if you look at the blow-up of the region down here, you see a number of genes, and you see that in this region in total, the serine codons that we wish to target ends up in a total number of 83. We're looking at 157 leucine codons and an astonishing number of 374 instances of the alanine codons we want to target. The strategy from here is to take this region, introduce synthetic DNA where all the codons in question has been replaced, integrate it into the genome, and see what the organism tells us.
The first set of codons we actually try to replace in this particular defined region is the serine codon TCG and TCA. We basically the first recording scheme we actually tested to replace the serine codon TCG TCA is recording scheme 1 as dictated by codon adaptation index. And basically what we did is we synthesized the entire 20 kb following this particular recording schemes and used Rexer attempting to replace the genomic fragment, the genomic 20 kb with a 20 kb synthetic DNA. However, to our astonishment, although the beginning and the end of this particular synthetic 20 kbs are successfully integrated into the genome, if we look into the middle of the 20 kbs, we always see a chimera between recoded sequence and wild-type non-recoded sequence. And these are all the positions of the attempted recordings in this particular 20 kbs based on the serine target codon TCG and TCA. If the recoding is successful at one given position, we color that position red and give the position value 1. However, if the recoding fails, basically the sequence remains as wild type in one particular recording position, we color that recording position black and give that recording position value 0. And as you can see, among all the individual clones we pulled out after Rexer for this particular recording scheme 1 for serine, TCG, and TCA, every single codon is a chimera. There's no fully recorded clones. And initially, we were very confused. And in order to gain a better understanding of the recoded versus non-recoded regions among the 20 kb sequence, we compiled all the individual clones. We compiled the sequence of all the individual clones into something we call the compiled recording landscape. in which, to our astonishment, we see there's one particular codon position that always fails to be recoded. Essentially, no matter how many clones we picked, that single position is always stays as wild type, can never be recoded into the AGT. And then we tried different recoding schemes. Recoding scheme 3, which is actually highly related to recoding scheme 1, which only differ in the recoding rule by one nucleotide. However, on the contrast to recoding scheme 1, recoding scheme 3 just works pretty much all the way through for all the clones. And essentially 88, almost 90% of all the clones are fully recoded. And this is really confusing initially for us, and we were wondering what's going on. And this is basically what we think is happening. If one particular recoding scheme, for example, in the case of recoding scheme 3, is fully viable, then the synthetic piece of DNA generated based on that particular recoding scheme is also fully viable. Under such circumstances, we can just very easily and straightforwardly replace a genomic fragment using the synthetic DNA right away, and basically having the final product with successfully replacing the 20 kb target regions with fully synthesized synthetic recoding sequence based on recoding scheme 3. However, if one particular recoding scheme is not so viable, like in the case of recoding scheme 1, then the synthetic DNA synthesized based on that particular recoding scheme 1 will contain a synthetic lethal, will contain a deleterious sequence. Under such circumstances, halfway through Rexer, you will have a genome that actually contains a synthetic lethal sequence, that actually contains a deleterious sequence, which will kill the cell. And now that we are only select for survival colonies, the only way for the cell to survive this is using the wild type piece of genomic sequence and to cross over and replace that particular synthetic lethal sequence and form chimeras between a synthetic sequence and wild type sequence. And in this way, essentially the cell can survive. And there would be many options to form such chimeras to enable the cell to survive. And taking advantage of the fact actually we have really high efficiency with the Rexer systems. We have like thousands to millions of such chimeras, of such chimera clones for the particular recoding scheme 1. And we can basically sequence them and compile them into the compiled recoding landscape, which will indicate precisely where this chimera always happens, which indicates precisely where the synthetic lethal is in the original synthetic DNA. So the Rexer system is not only able to allow us to differentiate between the viable and non-viable recoding schemes, but more importantly, it allows us to identify the synthetic lethals, deleterious sequences within the non-viable design of the synthetic DNA. And I mean, this is recoding scheme 1 compared with the other two recoding scheme 2 and 3 for the serine target codon TCG and TCA. Recoding scheme 1 has that synthetic lethal in that particular position, while recoding scheme 2 and 3 is fully working and fully viable. And basically, we identify that particular synthetic lethal based on Rexer. And then the question becomes, can we actually fix such synthetic lethals also using the Rexer systems? Especially if we know that particular position can be successfully recoded in the other two serine recoding schemes 2 and 3 using a different alternative synonymous codons. And the answer is yes. If we actually use the solution we find out in the other two recoding schemes to actually repair the particular synthetic lethals in the recoding scheme 1, then basically we can truly revive the entire recoding scheme 1 by just fixing one single nucleotide in a single codon. And this is allow us to basically say Rexer is not only able to replace big piece of genomic fragment using synthetic DNA, more importantly, it allows us to identify any potential design flaws within that synthetic DNA, and also allow us to very reliably and quickly fix such design flaws before we move on to the next step. So in this way, Rexer allows us the really unparalleled ability to very quickly go around the design, test, identify flaw, fix flaw cycle to actually debug any potential design flaws we may initially mistakenly introduce into our synthetic DNA design. This will allow us to very quickly walk along the wild type genome, making that into eventually a de novo synthesized one. And also, more importantly, on the way to this final goal, we will gain many valuable information regarding the genome design features. And here are all the 8 different recoding schemes we set out to test. And here are the results. I just want to highlight the alanine recoding scheme 7 and 8, although they target the same alanine target codons, GCG and GCA, and also although the recoding schemes also only differ by a single nucleotide. Recoding scheme 8 just fails catastrophically, while recoding scheme 7 pretty much works all the way through, replacing pretty much all the almost 400 codons all at once. And here are the 8 different recoding schemes we set out to test. And 3 of them works right away, while 1 requires 1 particular fix at 1 particular position. And after fixing, essentially all 4 recoding schemes works on that particular 20 kb region flawlessly and have no growth defect when compared with the wild type E. coli. So now we not only have the ability by the Rexer genesis to very quickly convert the wild type genome into a synthetic genome, we also have a design. And more importantly, based on Rexer, we have the ability to debug our initial design of any potential design flaws. So altogether, we really want to basically expand this principle and truly put the notion into action, and making the wild type E. coli genome into a truly synthetic genome to support a synthetic life form that fundamentally go beyond the limits of nature. And to this, we would like to thank our colleagues and comrades in this pursuit, in this seemingly crazy pursuit. Dr. Simon Brunner, together with us, developed all the different recoding schemes, and Sam Kim and Ting Sang Qiang, together with us, essentially implemented all the different recoding schemes and calculated all the compiled recoding landscapes. And we would also like to thank our supervisor, Dr. Jason Chin, for his trust and support, because initially the whole idea is just like one crazy idea on a piece of paper. And we would really like to thank Jason's trust in us to pursue this seemingly elusive dream. And we don't really know who is the crazy one, the one that has a crazy idea, or the one who actually believes the one who are crazy. Thank you.
Thank you so much, Dr. Wang and Dr. Fredens. This was very educational and very informative. I really enjoyed your presentation, and I'm sure our listeners as well enjoyed your presentation as well. So right now we actually have quite a few questions, so we will take a brief pause and collect all the questions, and then we will be back with the question and answer session. Thank you very much.
Thank you.
Okay, so the first question which actually we have, and that is something which we actually anticipated as well, is that how many days does it take to perform one step with your Rexer method?
Right, so basically the Rexer method is designed to basically follow a scheme of 7 days. The actual experiment actually takes 6 days, because on the 7th day we rest.
Thank you very much for that. And I also saw another question, which was actually talking about, during your presentation, you showed steps of 100 kb. And how do you reckon you can actually go for up to how much kb of sequences you can do?
Potentially, the technology is designed to be length dependent. And potentially, so long as we can keep the synthetic DNA stably inside the cell, it doesn't matter how long it can go. It can go potentially to megabases or tens of megabases. But practically, we're actually limited by the ability to actually easily keep the synthetic DNA inside the cell. Right now, we are using BAC, and based on the BAC, the bacterial artificial chromosome, we have reliably demonstrated it can at least keep 300 kbs. So I have to say the limitations is at least 300 kbs, and there's no upper limit. And right now, I understand in the paper we only published data for like 100 the replacement and integration. But actually, following the publication, so far we have demonstrated really straightforward replacement of like 180 kbs and higher in the lab. So with no deduction in reliability or robustness or efficiency. So we truly believe this is a technology that allows us to do integration and replacement regardless of the length, and but at least 200 kb, I would say.
Wow, that's pretty impressive, and thank you for your answer. The other question which actually we got is, so during your presentation, the work which you have shown is actually in E. coli, which is naturally recombinogenic. But can your Rexam method be extended to other organisms as well?
Yes. Actually, the Rexam method is designed to be compatible in all domains of life, to basically fundamentally overcome limitations of other methods, which is highly specific to a defined organism systems. And essentially, there are two main pillars to support the Rexam system to work. One is the recombination. The other one is the CRISPR-Cas activities. And we know CRISPR-Cas activities is functional in pretty much all domains of life. And the recombination activities actually is basically we rely on the lambda, right, such that we do not necessarily depend on the intrinsic recombination activity of a given host. But potentially, if you want to expand, say, for example, into mammalian systems or even human cell systems, then the system is actually naturally recombinogenic. So it is actually potentially even more ready to actually implement the Rexam system than bacteria. And actually, right now we have works undergoing right now actually to implement and expand the Rexam principles into mammalian systems.
Okay, thank you very much for that as well. And in the interest of time, we will actually probably have one last question. And that question is pretty actually interesting, but also I think it's a very popular question, I think. So how long will it approximately take to recode the whole genome?
Essentially that depends on how good the design is. And if we assume the initial design is flawless, then we know the equilibrium is around the size of 4 to 4.5 megabytes. And if we say conservatively we can replace the 300 kbs a step, then potentially we can basically do the entire E. coli genome within say around 15 steps. And each step takes like a week, then that's around 15 weeks. But this is assuming essentially do not need extensive fixation and repair of the initial design. But potentially for any initial design, especially for any first design of the synthetic genome, there are potentially many unknown unknowns, potentially many unknown mistakes. And we would probably be fixing all these potential mistakes on the way, which can slow down the process. And exactly how many such mistakes we're gonna encounter, that is totally kind of unknowns. We are trying our best to design the synthetic genome to be free of any synthetic lethals. But now that we truly think our understanding of the genome architecture and the codon usage is very limited, potentially on the way we're gonna really encounter many uncharted territories. And actually, we take this as one advantage, because then on our way to make the synthetic genome, eventually we will not only make synthetic genome, also on the way we will gain valuable and fundamental understandings about sample genome architecture and also codon choices. Hopefully 15 weeks, but maximally potential couple of years at least.
Okay, thank you very much for that. And so this is all the time we have today. I would like to thank Kai-Han and Julius for presenting as part of our GeneWest Week webinar series. Along with our webinar series, we have other ongoing activities such as a lab startup program, GeneWest Credit Awards, and exclusive promotions. Make sure you check out genewestweek.com.